A neuron is a type of cell found in your body that acts as a highway for information and sensory perception, helping you to see, hear, taste, touch, and think. Neurons are found all over your body, from your fingers to your eyes to the microvilli in your intestines. They connect organs to your brain, sending signals to and from your peripheral and central nervous systems. But how do neurons work? How do they share information? In this video, learn what the structure of a neuron is and how each of its pieces work together to make your brain and body function. Here we can see a model of a neuron's axon. The yellow foam represents the cell membrane that separates the inside and outside of the cell. A neuron spends a lot of time firing or sending information from different parts of your body, but it also spends some time resting. In this state, a cell has potential because there is a difference in charge between the inside and outside of the cell. If we consider the outside of the cell to be the ground, or 0 millivolts, the voltage of the inside of the cell is negative 70 millivolts. This difference in electrical charge occurs because of the ions and molecules that are found inside and outside of the cell. The inside of a neuron is full of potassium ions, a few sodium ions, and negatively charged proteins. Because the negatively charged particles outnumber the positive ones, the inside of a neuron is more negative compared to the outside, which also contains more sodium ions. These difference in concentrations and the resulting charge gradients are essential to the resting potential. Potassium and sodium ions move across the cell membrane using protein channels. Potassium moves via leaky potassium channels, which allows potassium to move outside of the cell down into its concentration gradient. These channels are specifically shaped so that only potassium can move through. But if these channels pull potassium outside of the cell, why are potassium ions still more concentrated inside? The sodium-potassium pump is a channel that pumps the potassium back into the cell, maintaining the concentration gradient of potassium. The sodium-potassium pump also manages the concentration of sodium ions. The pump moves sodium ions out of the cell so that the sodium remains at a higher concentration outside of the cell. The pump functions by using ATP, a form of cellular energy. ATP binds to the pump and gives it a phosphate, changing the shape of the channel to allow it to transport ions across the cell membrane. There are also gated sodium channels that remain closed during resting to prevent the sodium ions from moving across the membrane. The leaky potassium channels, the sodium and potassium pump, and gated sodium channels maintain the charge and energy potential of a resting cell. But a cell resting isn't the most exciting part. The cell transmits information through an action potential. When a stimulus triggers an action potential, gated sodium channels open, allowing some of the sodium outside to enter the cell and reduce the concentration gradient. The increase of positive ions inside the cell raises the voltage of the cell to reach a threshold of about negative 55 millivolts. This process is called depolarization. A neuron is able to reach the threshold of negative 55 millivolts with a strong enough stimulus. This threshold triggers even more sodium channels to open and raise the voltage to a peak of about 40 millivolts. The opening of sodium channels continues down the axon, and the action potential can travel down the neuron. The neuron has to return to resting state to fire an action potential again. Once the peak voltage is reached, gated sodium channels close, stopping the flow of positive ions into the cell. Gated potassium channels open, allowing positively charged potassium ions to flow out of the cell, and the charge of the inside of the neuron begins to drop. This process is called repolarization. Because the potassium channels are slower to close, the cell actually becomes hyperpolarized, meaning its voltage drops below negative 70 millivolts. Eventually, resting potential is restored thanks to the potassium-sodium pump, which pumps sodium ions out and potassium ions in so that the cell can be ready to fire another action potential. An example of action potential is the cockroach lab. During this lab, electrodes are placed into the leg of the cockroach and connected to a spiker box. The box reads the electrical signals that travel down the leg when it is touched. As the leg is touched, the phone shows spikes, which are the action potentials of the sensory neurons. These action potentials relay the sensations of being touched to the cockroach's brain. What we see is the electrical spike of depolarization of the neurons. Some of the neurological processes we've learned about can be stopped or inhibited by proteins or chemicals that are better known as neurotoxins. One such example of a neurotoxin is batrachoxin, commonly found in poison arrow frogs, as well as the pichu bird, found in New Guinea. The poison can be found in the bird's feathers and skin and was discovered when researchers handling the bird found their fingers going numb. Batrachoxin works by opening sodium channels of nerve cells and preventing them from closing. This leads to paralysis and eventually death. Batrachoxin is the neurotoxin most commonly found on darts and poison arrows used by indigenous tribes and also mimics the effects of the foxglove plant on the heart. It is possible in the future that batrachoxin could be used as an ingredient in painkiller ointment, but as of now, its healing powers remain elusive.